Bioinfocongress IV. Associate Professor Dr. Serdar Bozdağ. Integrating Multi-Omics datasets using graph representation learning. Associate Professor Dr. Serdar Bozdağ received his bachelor degree in computer engineering at Marmara University and Doctor of Philosophy degree in computer science at the University of California, Riverside. After a postdoctoral fellowship in National Cancer Institute at the National Institutes of Health. In 2012, Dr. Bozdağ joined Marquette University as an assistant professor in the Department of Mathematics, Statistics, and Computer Science. In 2015, he received the Way Klinkner Young Scholar Award. In 2019, he received the NIH prestigious Maximizing Investigators Research Award, a 5-year single PI research grant. In 2020, he joined University of North Texas as an associate professor in the Department of Computer Science and Engineering and the Department of Mathematics with an affiliation with the Biodiscovery Institute. Dr. Bozdağ has served as a program committee member in several bioinformatics and machine learning conferences, including ISMB, KDD, BIOKDD, BIBM, and BCB. He is an editorial board member of Plus One and Cancer Informatics journals. He has been co-organizing the annual international workshop on high-performance computing, big data analytics, and integration for multi-omics biomedical data since 2020. Dr. Bozdağ's research goal is to develop open-source integrative computational tools to analyze high-dimensional biological, clinical, and environmental exposure datasets to infer context-specific gene regulatory interactions and modules, and to predict disease-associated genes and patient-specific drug response.
Dear Associate Professor Dr. Serdar Bozdağ and participants, welcome all of you to our Bioinfocongress IV event. Dr. Bozdağ will give us a talk today on integrating multi-omics datasets using graph representation learning. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Bozdağ, welcome back. Thank you for being here.
Thank you. Thank you for the invitation.
If you wish, you can start your presentation.
Absolutely. Thank you for the organizers for inviting me to this wonderful program. I checked yesterday's videos, really wonderful speakers, wonderful talks. So I will be talking about some of our recent research on integrating multi-omics datasets, particularly using graph representation learning today. So let's get started. So as you all would know that human genome sequencing project was one of the important, very crucial milestones in the recent history for biomedical science. The project itself started in 1990. It took 13 years to complete, a little less than what they anticipated in the beginning. The cost was about $3 billion. Nowadays, we can do the same thing in a couple of days, and it would cost less than $1,000. So it's a great improvement, achievement for the biomedical field. With that technology development, lots of data sets have become available. Many international consortiums have been established to generate many different biological big data sets, from gene expression to protein expression to transcription factor binding sites lots of different data sets are publicly available. Anyone could download and utilize those data sets, those huge data sets. Most of those data sets are based on the central dogma of the molecular biology, where we try to understand the protein activity from the DNA data. So the transcription, at the transcription level, we look at the gene expression, and from the gene expression, we predict the activity of the proteins, because proteins are the building blocks in our bodies. They are involved in many functions. So to understand the gene activity is really crucial. So most of those data sets, you'll see that gene expression data is generated as a proxy of the protein activity. Gene activity is crucial because it's been already established that the gene activity is tissue specific and also disease specific. If you look at if you collect different RNA from different tissue, look at the RNA abundance, gene expression, you could be able to differentiate what tissue it came from. You could also look at the data from two types of cancer, or cancer versus normal, or one disease versus another disease, and look at the gene expression data. You might be able to distinguish the diseases as well. So our group and others have utilized such data sets to build predictive models to better stratify patients into subgroups, like glioblastoma patients, brain cancer patients, into different subtypes based on their gene expression value, as well as their DNA methylation activity. So all of those are great, but when you look at only one data set at a time, you might be able to mischaracterize the disease or the current underlying biology. To be able to really understand the whole biology, or at least move towards to that direction, it's crucial to look at different data sets simultaneously and utilize the complementary effect of those different data sets, what they tell us. Otherwise, we may not really fully characterize the elephant in the room. Like one maybe striking example for that is I'm going to give about the glioblastoma example, GBM. So back in 2009, the Cancer Genome Atlas group published their landmark study on GBM gene expression data. So they've done unsupervised analysis and came up with those 4 subtypes of GBM. So those 4 subtypes are proneural, neural, classical, and mesenchymal, and this is the gene expression heat map for some number of genes. And we just see that there is a distinct separation between them based on their gene activity. A year later, so particularly let's focus on the proneural group, because a year later, there was another study based on the DNA methylation of the same cohort, GBM patients. Now we do unsupervised analysis. We cluster the GBM patients based on their DNA methylation. And this time, 3 groups emerge, particularly one group, this is called GSIMP positive group, and the GSIMP negative group. That this group is being shown to divide the proneural group into 2 subgroups. So the proneural group is the group over here. Based on the gene expression, they look pretty homogeneous, but based on their DNA methylation signal, there's a striking difference between them. So some of the proneural groups are in this blue group, some of them are in this red group. And when we look at the survival data for those patients, so there are 20 GSIMP positive group and then 49 GSIM negative group, and this is the blue one is the non-preneural, the black one is the non-preneural group. Now, when we look at the survival difference, so this is the survival curve. For those of you who are not familiar, x-axis is the time after diagnosis, and y-axis is the probability of survival. In the beginning, everyone in the cohort is alive, so we are at the 100%, and as time passes, the survival rate decreases. And in the blue group, within 240 weeks, almost the entire population passed away. Whereas in the red group, they are still alive. So the red group is doing much better survival compared to the blue group. And again, based on the gene expression data, we cannot really understand this difference. So utilizing more data modalities is the crucial thing. So I'm gonna use multi-omics data sets. So I wanna just briefly tell you a little bit about those data sets. So what we talk about DNA methylation, which is a epigenetic signal, basically, in our genome, meaning that there is no change in the DNA, but there is a chemical change, especially in the cytosine. There's a methyl group added to the cytosine nucleotide, and because of that, that nucleotide is methylated, and this methylation has a huge impact on the activity of the genes. When we look at the gene expression versus DNA methylation correlation, there is usually a negative correlation between methylation and gene expression, meaning that when methylation is up, gene activity around that area will be down. Another omic data is structural changes in the DNA. These are not unlike DNA methylation. It's not epigenetic. It's a genetic change. It's either amplification, a portion of the DNA is amplified, so there are multiple copies of that, or it's deleted, so that copy is lost. Usually we have 2 copies of a gene, but due to copy number changes, we might have more or fewer copies. So this is also another indicator of gene activity. There are lots of biomarkers associated with different diseases based on the copy number data. Another one is microRNAs. These are short non-coding RNAs, and previously they were thought, oh, these are just junk information, not useful. But then it's realized that microRNAs actually bind to the mRNA, especially the 3' UTR of the mRNAs, and they inhibit their translation. So mRNA is not translated to protein through translation due to microRNAs. So microRNAs also have been shown really involved in several normal physiology as well as diseases like cancer. There are several onco microRNAs and also tumor suppressor microRNAs available. So utilizing microRNA data is also a crucial thing because it tells us something additional, some additional information about the underlying biology. So maybe in 1990s, the biggest bottleneck was to generate the data. It was taking years, billions of dollars to generate the data. But nowadays, that bottleneck is no longer exist. Data generation is relatively easier, faster, and cheaper. The new bottleneck, in my opinion, is on the computational side. All those data sets, how we're going to analyze, integrate all those data sets to generate some interpretable results, not only like some predictions where nobody knows why this prediction is made, but also interpretable so that it can be also applied in the clinical setting. Epigenetic data, RNA expression data, structural changes data, as well as the collective knowledge base that is being growing every day: gene ontology, protein-protein interaction, pathways, lots of annotations about drugs, diseases, phenotypes. All of those could be added together as multi-layered approach: genome, epigenome, transcriptome, proteome, metabolome, and could be able to utilize in various aspects of biology, particularly precision medicine, where the goal is to come up with individualized treatment options based on their based on people's individual data, not only coming from the multi-omics, but also environmental exposure, like the diet, lifestyle. This kind of information could be also added to the ingredients of lots of other data sets to be able to better build predictive models, better clinical decisions. So to this end, I want to tell you that in our lab, we are dedicated to this problem: how we could come up with computational tools that will combine different data modalities. I would say that at the core of our lab, we do machine learning, deep learning for various problems. Like for several of the cases, we do multi-omics data integration, and we do work all the way from at the cellular level, trying to understand gene regulatory networks for different contexts, for different conditions, as well as at the population level, some work on precision medicine. So today I'm going to talk about at the population level type of work, where we've come up with a method to do cancer subtype prediction, integrating multi-omics data sets. And for machine learning, we are actually utilizing a graph representation learning method called graph convolutional neural network. So this work is available on bioRxiv as a preprint at the moment, and it's under review at a journal. The code is available on our GitHub page, and this work was mostly done with by my PhD student, Zinet Nesibe Kesimoğlu, who is graduating hopefully this summer. So I'm gonna give you some background on this work. I'm gonna share our results, and then I'll conclude and take any questions you might have. So as you all know, cancer is one of the leading causes of death, as mentioned in Sudraka Mukherjee's book, Cancer is the Emperor of All Maladies. The number of cases increase and is expected to increase, partly because there are more diagnostic methods nowadays, but also the economic burden of cancer is also huge. Among those, breast cancer is the most prevalent cancer type in the world at this time. There are different groups of breast cancer, and depending on those groups, the survival rate differs, the treatment options differs. So one grouping is based on the biomarkers, estrogen receptor, progesterone receptor, HER2 protein. So if those receptors or protein abundance exist in the patient, then there are certain treatment options that would target those receptors. But there are also cases where none of those are positive, so it's a triple negative breast cancer, which is more aggressive type of breast cancer. But still, when breast cancer is diagnosed early, the survival rate is way better compared to 15, 20 years ago. So this is based on the immunohistochemical data, but also there are molecular subtypes of breast cancer. So based on the gene expression data, again, we're coming back to the gene expression data. As I said in the beginning, gene expression data can be a good data set to characterize the diseases, and it was easier to generate as well. So for many diseases, for many tissues, gene expression data was generated. That was one of the most commonly used, like omic data, transcriptomic data. So in 2009, Parker et al. Identified 50 genes out of 20,000 plus genes in human. Those 50 genes were identified to cluster breast cancer subtypes into 5 groups. So in the heat map, we see those 50 genes and those 5 subtypes: luminal A, luminal B, normal-like, HER2-enriched, and basal-like. So this is totally based on the gene expression data, and based on those 5 subtypes, there are different characteristics that exist. In fact, the TCGA group, when they published their landmark study on breast cancer, they show for different data modalities what are the signatures for each of those 5 subtypes. So you see the 5 subtypes here, and then we look at the mutation data, clinical data, copy number data, and look at the differences between those subtypes. And this is also their clustering and how that clustering concords with the PAM50 clusters. So based on all of those, like we have not only gene expression data, but different data modalities for breast cancer, we ask, can we utilize those other data modalities to predict cancer subtypes? And the other question is, okay, if we could do that, because more data might give us more information, how are we going to integrate those multi-omics data sets was the next obvious question that we had to tackle. So multi-omics data sets and clustering based on multi-omics data set is a really popular area. There are several methods on this, like iCluster. iCluster Plus is one of the methods where the goal is, by applying dimensionality reduction on different data modalities, trying to make the data sets align at the latent space. Similarity network fusion is doing a different approach, building patient networks, patient similarity networks for each of the different data types, and integrating, merging those networks by applying a nonlinear fusion step. On the other hand, PINS Plus is also another clustering method, and the intuition behind this method is that if 2 patients are in the same group, so they are in the same subtype, then if I perturb their data slightly, still they should end up in the same cluster. So they do perturbation on the data and check how stable the clusters are. If certain samples end up being in the same cluster through those perturbations, then they end up being in this same cluster in the final results. So like in CINEF, graph is a natural data structure to represent biological data sets. So we see lots of examples of that in biology. We look at the gene regulatory networks, we look at the protein-protein interaction networks, we look at co-expression networks, we look at gene ontology. All of those different data modalities are represented as graphs. Once we have the data as a graph, then we can basically identify communities. We can identify nodes with too many connections, so which could be maybe the important nodes, important genes, important terms. And we could also identify different groups that work together potentially. So what we want to do is a little different than that. We would like to build those graphs where each node is a patient. But our goal is, what is the subtype, breast cancer subtype in this particular example, of that patient? We would like to make a prediction. So it's not unsupervised analysis, it's a supervised analysis. We want to do a prediction. But how am I going to do a prediction on a graph data? Graphs are not Euclidean space. For a graph, there is no ordering. For a graph, there is no fixed features. So I need to do something, and this is called graph representation learning, node embedding, network embedding, graph embedding. There are different names for this, but the idea is each of the nodes, can we come up with a vector representation? Just a bunch of features, maybe 100 or 200, maybe 500 features, where the similarity of those vectors in this embedding space will reflect the similarity of the nodes, like they are close to each other in the network. So they have similar topological structure, or they have similar labels. So from the graph data, come up with an embedding where the similarity in the embedding will reflect the similarity in the graph. And once we have that embedding for each of the nodes or for the entire graph, then using machine learning methods will be straightforward, because it's just going to be like a tabular data now. So we could do node classification, we could do link prediction, we could even do community detection again, like applying clustering methods on that embedding data. All of those are possible. So in the recent years, these methods have become extremely popular because graphs are great to basically load lots of different data sets. And then from graph to machine learning tasks, we need this graph representation learning step to move forward. So there are two streams of work in this domain. One of them is the so-called shallow network embeddings, where this is mostly based on a random walk approach, but there are also some matrix decomposition based methods as well. And there is deep learning based methods, which are collectively called graph neural networks. And that's what I'm going to talk in more detail in the next couple of slides. So this part of the talk is a little bit technical. For those of you who are interested in this concept, I wanted to add these details. I also put some links to some tutorials. So Leskovich group generated really nice courses and tutorials on this. So if for those of you who are interested, I would highly recommend. So to be able to do graph embedding using deep learning, so graph neural networks, what we start with, this is like a very high level picture. Basically, what we see is the graph is an input, and then we do some type of graph convolution to gather information for the neighborhood of each of the nodes. That information gives us some feature, some initial embedding. And this embedding is passed through an activation function, goes to another layer, and in that layer, maybe we'll do some regularization like dropout. Maybe we'll have another convolution layer, and then at the end, we have this output layer where we have the final embeddings. So if you are familiar with neural networks, this is the same idea. It's just how we define the graph convolution is the part that we need to figure out. But the rest is very similar to the neural network approach. So the graph convolution thing is based on two things. One step is we're gonna define the neighborhood of a node, and we will do the aggregation from those neighborhoods. And how we're gonna do this aggregation, are we gonna do the sum or the average? Those are some of the questions, and that's how the different methods differ. So here in this toy example, we have a node A. I want to learn the embedding of this node. And to be able to learn the embedding of this node, I'm going to look at the neighbors of this node. The immediate neighbors are B, C, and D. So I'm going to get the embedding of B, C, and D, aggregate them, will feed that information to a neural network, which will be trained. And that through that process, I'm going to get the embedding of A. But this is a two-layer model in this example. So two-layer meaning a B also gets the embedding from its neighbors, which is A and C. So the information comes from A to C to B, and from B back to A. So in a two-layer approach, we get the neighborhood. So for instance, A that way gets the information from the two hop distance. It gets the information from the immediate neighbors, B, C, D, but also the neighbors of the neighbors, for instance, F and E, and collect all this data and come up with the final embedding. If you go a little deeper to the math, this is the idea. And again, if you are familiar with the neural networks, this formula is also not too complicated. So we have a multi-layers in this example, 2 layers. Here, K is the total number of layers. Based on the previous layer, we look at the information from the neighbors in the current step, the neighbors. We look at the embedding of the neighbors. We average that embedding, and we do a linear transformation on that embedding, and we add it with the current embedding, and everything is passed through a nonlinear activation function, and that will give us the embedding of the K+1 layer. And we continue like that. At the end, we get the final embedding. So initially, we have some node features. Those node features will be our initial embedding. And with that initial embedding, we do embedding at layer 1 and embedding layer 2, layer 3. Mostly we do 2 layers to avoid some over-smoothing issues. One of the popular methods is graph convolution networks, and this whole process is summarized into this formula, where H is what we are learning, the embedding. So this is the embedding at layer L. A is the adjacency matrix to represent the graph, and D is the degree matrix. Again, this is also coming from the graph. So H, in the beginning, we have some initial node features that we use as embedding. So all of those are coming from the graph. And the W is what we are learning, the learnable parameter. And sigmoid here is just an activation function like ReLU or sigmoid or softmax, whichever is used, but mostly ReLU is used. Okay, so I'm just gonna, so this is kind of concludes the technical part. So there are some graph convolutional network-based methods on omics data. GCNC, MogoNet are some examples, but there are some limitations. So they use single graph or limited omic data, and they don't analyze multiplex networks simultaneously. Like they analyze one network at a time, not like in the network, there's only information about one data modality. So to address these limitations, we come up with Supreme subtype prediction methodology. Supreme is a node classification framework. We encode multi-omics features and then we do convolutions on multiple graph structured data. Again, convolutions on multiple networks. We utilize features and associations from multiple data types at the same time. And there's no limit on number of data modalities that you can utilize. So here's the framework. And I also want to say that this is a generic method, not cancer or even not related to biological data necessarily. As long as you have data from different data modalities for some entities, then you can have graphs, and then you can learn the embeddings of those graphs using Supreme. So here's the idea. So let me check the time. Okay. So we basically built patient similarity networks, multiple patient similarity networks for each data modality, let's say gene expression or clinical data or co-expression data or DNA methylation data. For each of those data modalities, we built a patient similarity network. And for each network, here the nodes are patients, and the nodes have some features. So those features are also features from all those different data modalities, all combined. So the topology of the network is based on a single data modality, let's say gene expression. But the features are from all the data modalities. So for all data modalities, we have those networks generated. Then we apply graph convolutional network on each of those graphs separately, and we get node embedding for each of the graphs. And the nodes are the same. They are the same patients. So all networks have the same nodes, but the topology is different. Features are also same because they are coming from all data modalities. So we learned the embeddings. After we learn the embeddings, then we say, okay, we have, in this example, 7 types of data modalities. So we don't know which of those will be most useful. So what we're doing all combinations of them. For all combinations, 2 to the 7 minus 1 cases, we integrate those node embeddings. And optionally, we also integrate the features that we started with over there as well. I mean, they were using the graph, but maybe that information got diluted. So adding this feature is also another option. And then after that, we do the prediction. For prediction, we use any machine learning task. In our example, we use multi-layer perceptron. And the final outcome is the subtype of this patient. Basal-like, HER2-enriched, luminal A, luminal B, or normal-like, which is the PAM50 subtypes that I mentioned 10 minutes ago. Okay. So we use data from TCGA, the Cancer Genome Atlas, and Metabrick databases. We use several data modalities from those data sets. Metabrick didn't have all those data modalities, so we use fewer data modalities for that one. And we also combined these 2 data sets and did another analysis. So we extracted features, and selecting the top features based on the Baroda R package. We z-normalize all the features and combine them and use them as node features. To generate the networks, we use different similarity metrics like Pearson, Jaccard, Gower, based on the data modality. If it's a binary, then we use the Jaccard index. If it's gene expression data, then we used Pearson correlation. We use the top ads as the patient network. So I'm not going to go into too much detail, but these are some of the things that we had to play with. So we tried different values, how many gene ads to keep. And so we then finally we fixed that number through hyperparameter optimization. And again, features from all data modalities are added as node features. So here are some results. So first of all, some information about our data. As I said, we use the data from TCGA and Metabrick, and we also combine them. These are the number of raw features that we use. 29 features from clinical data set, 500 from copy number, 16 co-expression, 1,000 genes from expression data, 1,000 probes from methylation, 343 microRNAs, and 200 mutations. And for Metabrick, the numbers were more or less same. And these are the number of samples. So we have slightly over 1,000 samples in TCGA and almost 1,700 samples in Metabrick. And these are the groups of samples in each of the subtypes. So as you can see, most of them are luminal A. So the subtypes are not like balanced. So that's why in our metrics, I'm going to show F1 score, which is more reliable for unbalanced cases. And the proportions are more or less same for the Metabrick. Again, luminal A is the most prevalent group. Then we have like normal like is the fewest case. And then we combine all those data sets together to generate this combined data set. Okay, so remember, we try all combinations of all data modalities. So we have a bunch of results, and this violin plot illustrate those results. And based on these results, we were able to show that Supreme outperformed all the baseline methods and state-of-the-art methods like DeepCC and MogaNet and GCNC. And the baseline methods like random forest, support vector machine, XGBoost, and multilayer perceptron. For those baseline methods, we basically use the features and then do the prediction. Here, we're using the features as well as the node embeddings. And here, Supreme minus, we are only using the node embeddings. So the features are not added at the end. If you remember, I mentioned that we might add the feature as an option at the very end. So this Supreme minus is the one without those features added. And on TCGA data, we were able to show that there's a statistical difference between Supreme and other approaches. In Metabrick data, we were able to see the same trend. Supreme outperformed all the other methods. And when we combine, we were able to also show that there's like Supreme is doing better than the other methods. In this case, the number of data modalities is only 3, so there are only 7 runs. Because of that, it's hard to show statistical significance. So those values are not statistically significant, but that's because of the number of samples. The other thing that we've done is, okay, so we're doing this prediction. So we have 5 groups based on our predictions. Can we look at the survival differences between those 5 groups? And is there a significant survival difference between them? If there is, that's a good thing, because then our differences, our subtypes are meaningful subtypes. So here we look at the survival difference, statistical significance of the survival difference. So we get the p-values. Here, the lower the better, because it's more significant. And what we see here is Supreme and also Supreme minus, for most of the runs, got quite well separation. The other methods, there was significant difference with all the other methods, as can be shown here. Some of the methods actually got really high p-value for most of the cases. Some of them were still like giving low p-value, but still there was a statistically significant difference. Here, the solid red line is the 0.05 cutoff, and the dashed line is the the TCGA like the ground truth PAM50 subtype survival difference p-value. So this is the p-value that we get with the real data. So if you could zoom in to this plot, 0.05, 0.03 is what PAM50 is doing, and most of our runs are doing way better than even the ground truth. So again, I want to say what this implies. Like, we are making subtype prediction. If we do exactly same as PAM50, then our accuracy will be 100%, our F1 will be 100%, because we use PAM50 as the ground truth. But we are making some mistakes. Because of that, our subtypes are different, but those differences end up give us a better survival difference. So perhaps by using other data modalities, we are saying that a luminal A group is luminal B, for instance, and we also show that based on the survival difference, that our grouping is doing better. Like, this is the supreme subtypes versus PAM50 subtypes. We look at the Kaplan-Meier plot, the plot that I showed in the very beginning when I gave the TCGA GBM example. X-axis is the time, survival probability is the y-axis, and what we see here is we compute the statistical significance between the differences between those curves. So it's 0.01 for supreme, and it's 0.03, which is the 0.03 that you see here for PAM50 subtypes. It's hard to distinguish by eye, but it just says that the difference is higher for supreme case. We also look at the single data modality. So what if we only focus on one data modality, not all 7 of them? So it's just one run in this case, but we repeat that run multiple times, so we have some standard deviation shown here. So for most of the data modalities, Supreme did better than other methods, except for a couple of cases. Sometimes Supreme minus did better, but most of the time, adding the features at the very end showed us some useful feature. And we got this more or less the same trend for the Metabreak data as well. So in conclusion, I today talk about different data modalities. I talk about graph representation learning, especially deep learning on graphs, and discuss you about a tool that we recently developed, Supreme, which integrates different data modalities, and we show the results on breast cancer subtyping. Our results show that we outperformed baseline and state-of-the-art methods, also got significant survival methods. And this tool is publicly available. It can be applied to any other data type, disease type. It's very generic and user-friendly. With that, I like to give acknowledgement to my students, particularly Zinet, who did most of this work, and our funding agency for their funds. I'm really grateful to be working with a wonderful group of students. So we do lots of research, but from time to time we also have fun together. Next week, for instance, we're gonna have another lab lunch to celebrate Zinet's graduation. So she defended her dissertation successfully 2 weeks ago. So we'll celebrate that. So she will be joining NIH as a postdoc soon, hopefully. With that, I also would like to tell that we are hiring. My group is hiring postdocs and PhD students to work on those projects. We are in Dallas, Dallas Fort Worth Metroplex area. It's a wonderful place. It's a extremely growing area. The weather is beautiful. Schools are great. Like if you have kids, there is direct flight to Turkey and many other places in the world. And UNT is a R1 university, a research intensive university. So if you're interested, feel free to email me. So I put my contact information over here. Thank you so much. So I'm happy to take some questions.
Thank you so much, Dr. Bozdağ. I am directing our first question. In future, can Supreme be utilize attention mechanisms which allows getting weighted contribution from different data types and also weighted neighborhood from networks?
Wonderful question. So I'm really happy to hear this question. Actually, this is what we're working right now. So probably in a couple of weeks, hopefully, we're gonna release another preprint where we do attention mechanism between different data modalities, as well as between different nodes. So at the node level and at the network layer level, we compute attentions and we do the integration based on that. So we have some preliminary results. Hopefully, we're gonna make it available really soon.
If you'd like to, you can close your presentation and see the questions yourself.
Sure.
Okay, thank you. Thank you for the response. I'm moving on to the next question. GNNs learn a node's features by iteratively propagating information from the neighboring nodes until convergence. They have high computing costs and the learning filters lack of the localization attribute. Is there any solution?
So, right. I'm not sure I fully understood the question, but yeah, the cost of analyzing GNN is out there. So there are some scalable GNN methods that could be used there. So there are some papers, so like published from different groups, like Leskovich group recently published a work on that as well. So there are some approaches. So to be able, so first of all, so when we run, our most of the time we spend is on hyperparameter tuning. There are some parameters that we need to optimize, and for that we have to run many times. Each run is not taking lots of time for us, but again, our graphs is relatively tiny. But if there's like, let's say graph like a social network where there are millions of nodes, then this becomes there's a scalability issue. But yeah, there are some work on that domain to address this issue, but still it's an ongoing research problem.
Thank you for your response. I'm moving on to our next question. Omics are heterogeneous entities with different data distributions or data types. How is it possible that these inconsistencies between omics do not hinder their integration?
Oh, it does. It does. Like if you simply say that, okay, let's aggregate all the features all together and then do the prediction, more the merrier. It doesn't work that way. If we force that these omics data sets had to like align with each other, then that's also another problem, because as illustrated in this question, there could be limited data, or there could be some bias or noise in the data. And if we really force that all those data sets need to agree with each other, then we might be able to lose some of the some of the signal. So I think the key thing is, and this is a research problem that is not fully resolved yet, but I believe that there needs to be a trade-off between, like, at some point we need to say that this particular data piece coming from this particular data modality is not contributing, it's not agreeing with the others. So there is a higher chance that this is not complementary but noisy. So we should eliminate. But how to do that automatically in a customized manner, that's difficult. And again, that's why this is an active research area. Hopefully down the road there will be better solutions.
Thank you for your answer. We have a question from YouTube. Can self-supervised graph representation learning integrates multiple molecular networks and decodes gene-disease relationships?
Yes, it does. So so graph representation learning is a pretty custom method. So there are different downstream things that can be done. So this is also another work we're interested in. So we publish a work which is not using deep graph representation learning, but shallow learning method for this problem, like gene-disease relationship, in a sense. So once you have multiple molecular networks, let's say a gene network and a disease network, and even the gene network could be multiple layers, the disease network could be multiple layers between different disease ontologies, then you can define some tasks that tries to be able to do the self-supervised learning. You need to come up with a task for the program to learn by itself. And that task could be just based on the topological similarities of the nodes. For instance, sometimes this gene network graph is perturbed, so you have a different graph, and you try to say that the node embeddings that you learn will be similar in the real graph compared to the perturbed graph. Or there are some other tasks that you can come up with. After that, once you learn the embeddings, then to be able to do the link prediction, what people usually do is the embeddings of these two nodes are passed through a multi-layer perceptron, for instance, and then you do a machine learning, or you do dot product between these two embeddings, and based on that score, you say, okay, there's a link between this gene and disease or not.
Thank you for your answer. We have another question from YouTube. More data brings with it more information, more noise, redundancy, and increased computation time. How do you overcome this problem in multi-omic integration?
Yeah, so it's, again, there are different paradigms for multi-omics integration. So it's a really hot area. So we've been organizing workshops for the last few years on this topic, and many other groups are also working in this area. So there is not a single solution, but there are different approaches. Like there is early integration approach where all data sets are integrated in the beginning. There is late integration approach where data is handled separately and you make some final outcome or generate some type of process data from each data modality and then fuse them later. Like our Supreme is a late integration example. There are also hierarchical integration approaches where some data modalities are known to be related to each other, like utilizing this biological knowledge into the integration process. And completely different from like graph representation learning paradigm. Several years ago, like we've done this, and many other groups also have done that. Like some omic data sets could be utilized to generate some prior knowledge, prior network, and then using that prior information on a Bayesian approach could be another method for the multi-omics integration. But but I don't have a final answer to this problem. Again, today we're working on this in the maybe 10, 15 years, we'll have a better understanding on this, because the data noise will hopefully reduce, and we'll get more data modalities, and we'll have a better methods to handle these issues.
Thank you for your response. Another question from YouTube. Since graphs are formed based on samples rather than features, the complexity of the whole pipeline does not significantly increase by adding new omics modalities. Is this an issue on data?
So for this particular work, our grant truth came from the gene expression data. Remember, the PAM50 subtype labels are based on the gene expression data. So we've done great when we included gene expression features to the results. But again, that makes sense because the data, the grant truth, is based on gene expression. When we didn't have that, even we have other data modalities, we were not be able to achieve extremely high performance. I think this case is not extremely clear whether adding more data modalities are improving the results or not, because there are some ablation studies that we've done. I didn't mention in the presentation. For some omic layers, if we remove them, we didn't see much difference in the performance because the other data modalities were able to handle it. But then some omic layer, especially I remember copy number, for instance, when we don't have the copy number, then we had a significant drop in the performance. So yeah, there are some mysteries as well, but I think there are more tests that need to be done to be able to say whether certain data modalities is always useful or not useful at all.
Thank you again for your response. I'm moving on to our next question. Why do we not pursue an easier strategy than unsupervised learning? That is to train an end-to-end GNN in semi-supervised fashion.
Yeah, so we wanted to make this method as a generic method where the goal is to learn the node embedding. What you do with those node embeddings could be differ. But if you have a specific problem, yeah, end-to-end model could have been done. So our approach is in two phases. We learn the node embedding, and then the next problem becomes the machine learning problem of subtype prediction. But this was intentional. If we had the goal of just subtype prediction, we could have made the model end-to-end and like the back propagation goes all the back to the beginning of the pipeline. Yeah, this could be done definitely.
Thank you again for your response. Another question from YouTube. Could tasks such as the creation of multi-graph representation learning-based ensemble models and prognostic models and molecular subtypes for specific diseases become easier in the future?
Yeah, I think it will be easier in the future. Like the history taught us that as time passes, we learn more and more things, that we end up with more difficult problems to tackle on, but the problems that we work on today will be relatively easier down the road, because there are, like when we started this project, back then we didn't like this project started many years ago when there was not even graph representation learning. We've tried different machine learning methods in the beginning. Then we switched to graph representation learning. But then when we started, we used GCN. And after that, attention-based methods emerged, and many other advanced methods emerged. So yeah, the field is moving really fast. And if you look at some machine learning conferences, this is one of the most popular areas. Every day there are really high quality publications. So yeah, I hope that the problems will be easier and easier. But come up with new problems to work on, which will be the next level.
Thank you for the response. Another question from YouTube. There are some regulatory relations, such as competing endogenous RNA regulation, which has been recently discovered with important insights into cancer. To adopt this kind of regulatory relations, can Supreme be used?
Yes, actually, Zinet worked on this problem, cRNA interaction, as her first project, and that's one of her future goals as well. Like, can we utilize this information based on the cRNA network? Can we get another omic layer to this? Maybe whoever asked this question, Sedanur, maybe you read the paper, but yeah, thank you for this question. Absolutely, this is what we are thinking at the moment.
Thank you for your answer. Another question from YouTube. When conducting multiple omics experiments, collecting large amounts of data can often be done on a small sample of patients. Wouldn't this cause datasets with a number of variables exceeding the number of samples and machine learning algorithms to overfit high-dimensional datasets?
Yeah, this is a common problem that we face. So here we start with about 1,000 samples, which is relatively high compared to many different examples. But then if you look at all of our features, they are in tens of thousands of features. So to address this problem, we did feature selection. We reduced the features to a more manageable number. So there were about 4,000, 5,000 features at the end for 1,000 samples. And we check for overfitting. So we look at the performance on the training data and the validation data and the test data. And there was not much overfitting in our case. Thanks to the feature reduction, I mean, if you use all the features, definitely overfitting is a inevitable problem.
Thank you for the answer. Another question from YouTube. One of the toughest problems with neural networks is their black box structure, i.e. their non-interpretability. Is having a good predictive model enough for them to be interpretable?
No. So definitely there are some additional work need to be done to be able to better understand what part, what data modality, or even within the data modality, what subset of features are contributing most to the overall prediction. So there are several methods for this. Like, for those of you who are interested in this domain, there are explainability methods for deep learning methods in general. Like deep lift is one of them. Or GNN explainer is another method. So there are knowledge distillation methods where you try to build a simpler model to mimic your complex model. And this simpler model, like maybe based on decision tree, will be more interpretable. And so you could do this. So this is doable. We've done ablation studies to show the impact of different data modalities. But yeah, more things can be done to better interpret this complex model.
Thank you for your response. Our next question is from YouTube. In your presentation, datasets show different numbers of samples and results. How we can decide which dataset is more accurate information?
So the TCGA data is more comprehensive, so they have more data types, but the number of samples was fewer. I mean, it's hard to say which one is more accurate. All of them do rigorous pre-processing to make sure that the data sets are accurate, and they eliminate all the samples that they don't pass the quality control protocols. So we've tried those data sets separately, and we also combine them to look at the results, just to see if what we are getting is data specific. That was one concern we had, like we're getting those results because our model fit to this particular database or not. So we were happy to see that we were able to show performance similar like trends in two different data sets. So, I mean, the answer to that question, we don't know which data is more accurate or which data set have the most accurate information. But yeah, we use all the available data that we could use.
Thank you for your response. We have another question. A multi-graphical representation learning-based ensemble models are being developed to predict potential gene disease by extracting gene disease associations such as microRNA diseases, protein diseases, and gene pathways. Do you think that a similar path can be followed to improve the prediction of gene disease associations?
Absolutely, and there are right now I don't have any tool or paper to give on top of my head, but there are several studies that utilize this information, like represent different interactome and different layers of information, and to make those associations for sure. For as well, gene-drug interaction is also another domain. The similar idea could be applied to find associations between drugs and their target genes.
Thank you for your response again. I'm directing our next question. Mixed integration transforms each omics dataset independently. Much of the large matrix resulting from early and intermediate integration is not utilized by the machine learning model. So how do we determine which approach to use for multi-omics integration?
Yeah, very nice question. I don't know a definite answer to that. In my opinion, early integration is too simplistic, and late integration could be also potentially too late, meaning that if you just rely on three, let's say, data modalities, and then you just do the prediction of each of them separately, and if you look at the consensus, then you might have just by chance, we might have some mistakes, especially if some of the data modalities have some issues for some cases. So I would recommend, like, don't fix to a particular method, but try different approaches in the beginning, trying baseline methods. If you're able to get what you plan to get using baseline simpler methods, I would go with those. If you are in the computational science domain where your goal is to come up with new methods, then looking at the state-of-the-art methods, different approaches, and try to see under what conditions those methods might fail, might give you some hint about what would be the new novelty, new innovation in developing a new model. But if you're a user, like you have data and you want to know how we're gonna try it, simpler the better. I would say start with simpler models, and then if you are not happy with the results, go to more complex methods.
Thank you for the response. And moving on with the next question. Although there have been advances in causal modeling, existing approaches have difficulties in applying them to high-dimensional datasets due to the numerical complexity in the application of the approaches and the multicollinearity of the data. How do you think these difficulties can be overcome?
Wow, that's a really difficult question, really important question. Yeah, none of those things that we do have anything to do with causality, because I mean we're computing correlation. We do associations basically when we build those patient similarity networks. When we talk about causal interactions, it reminds me more like at the cellular level, for instance, gene regulatory network. Even for gene regulatory network, it's a difficult problem, because when you know that two genes have similar expression profile, those two genes might regulate each other, or they might be co-regulated by a third gene, a transcription factor, for instance. So it's really difficult. Based on the data, it's ambiguous to be able to say anything about causality. But if you use additional data, for instance, if there is ChIP-seq data, and if the ChIP-seq data gives us some idea about what transcription factor is binding to what gene, that could be one way to be able to define some directionality. Other knowledge bases, existing regulator target interactions could be another approach. And also in different areas of machine learning, there is like causal interaction inference methods exist. They apply to social networks, for instance, to be able to look at if certain tweet posts, for instance, influence certain actions. But for that, they have lots of data sets that they could use. For the biological data is a little limited. Another thing that comes to my mind is, if you have time series data, then you look at maybe the cause and effect over a time. But again, time series data is not available as much as the static data that we have. Yeah, it's good research area for sure, but the most of the issue comes from the data limitation.
Thank you for your response. As for our last question, what advice would you give to students studying in scientific fields?
Okay, wonderful. First of all, the questions that were asked, I'm quite amazed with the level of these questions. Wonderful. I'm really happy to again give this talk. I'm humbled. For scientific fields, like if we're talking about bioinformatics, I would say that let me step back actually. So when people tell me like what major to choose for their undergrad degrees, I usually tell them do interdisciplinary, like have maybe another major or minor, and like learning a foreign language, make sure to learn a programming language, whatever area you are in, biology, chemistry, journalism. Still, like learning computer language is gonna make it really useful, because data is becoming available everywhere nowadays. So to be able to handle this data, analyze this data, to be familiar with those methods would be useful. The other thing is just like attending these events are really useful. If you are a PhD student at that level, one recommendation that I would have is just make sure that you write every day, even if it's if even you don't have much to write. Still sit in front of the computer and like 15 minutes, not more than that, just write about your project, about your experiment, whatever you have, and that's just helps the brain to digest the data more, and that helps. Nowadays, blog pages and all the YouTube tutorials, they are really useful. So I learn a lot, and myself, and I would also recommend to utilize those resources and learning, to make learning as part of your routine. So writing, learning, and of course doing the research. I said I'm hiring students, so if you're thinking abroad, I would really recommend you to plan for that. Reach out to professors, read their papers, and ask them questions. And yeah, nowadays we're really, I personally have difficulty to hire good students to my group. It's very competitive. So from outside, it might look like that it's difficult to get into those positions, but actually, it's much easier if you make the first step. Yeah, thanks again for the kind invitation. It was wonderful to be with you all today.
Thank you so much, Dr. Bozdağ. On behalf of myself and all of our participants, we were so grateful to you. We are so grateful to you for spending your valuable time, transferring your knowledge to us, and for this informative presentation. Thank you.
Thank you. Bye-bye.
I wish you very healthy days.
Thanks.